Hello, I'm Zach Zuniga, and I will be covering cloning. What is cloning? Cloning is the process of producing genetically identical individuals of an organism either naturally or artificially. In biotechnology, cloning refers to the process of creating clones of organisms or copies of cells in our DNA fragments. There are three different types of cloning according to the National Human Genome Research Institute. Gene cloning, also called DNA cloning, creates copies of genes or segments of DNA. Reproductive cloning makes duplicates of whole animals. Therapeutic cloning creates embryonic stem cells, which are used to create tissues that can repair or replace damaged tissues. Why cloning is of interest. Cloning animal models of disease. Researchers can learn about human disease by studying animal models such as mice. Animal models are genetically engineered to carry disease-causing mutations in their genes. Cloning could help reduce the time needed to make a transgenic animal model, and the result would be a population of genetically identical animals for study. Make stem cells. Stem cells cloned from someone with a disease could be grown in culture and studied to help researchers understand the disease and develop treatments. Revive endangered or extinct species. A closely related species of an extinct animal that's currently living could serve as an egg donor and surrogate mother by using DNA from well-preserved tissue samples. Endangered species is much easier, mainly because the surviving animals can donate healthy living cells. Reproducing a deceased pet. If you really wanted to, and if you had enough money, you could clone your beloved pet. Don't assume your pet would be exactly the same. An individual is a product of more than its genes. The environment plays an important role in shaping personality and many other traits. Cloning livestock. Agricultural animals such as cattle and pigs are efficient producers of high quality milk or meat as well as breeding stock. Cloned cows can produce about twice as much milk as the average producer, and a cloned racing mule is ranked among the best in the world. Drug production. Scientists could take cells from a cow that produces large amounts of milk and grows them in culture. Then they could insert a gene into the DNA of these cells that codes for a drug or a vaccine. The nucleus is taken from one of these cells and transferred into a cow egg. It could develop into a cow that makes the drug in its milk. Every cell in the cow would carry the drug gene. It could pass the gene out to its offspring, creating a whole herd of drug-producing cows. We could avoid the issue of genetic reshuffling that happens during sexual reproduction and simply clone our drug-producing cow. Current state of cloning. 20 years after the cloning of Dolly the sheep in 1997, the first successful cloning of primates using somatic cells nuclear transfer was reported with the birth of two live female clones, crab-eating monkeys named Zhang Zhang and Hua Hua, the first match cloned by the Dolly method. The same researchers created the first cloned monkeys copied from a genetically modified specimen, which carries an altered gene related to the biological clock. This advance illustrates one of the greatest current uses of reproductive cloning, the creation of experimental animal models for study of diseases. Therapeutic cloning and reproductive cloning can be used for theoretical human cloning. Therapeutic cloning would involve cloning cells from a human for use in medicine and transplants, and is the active area of research, but is not in medical practice anywhere in the world, as of October 2019. There are currently no federal laws in the United States for ban cloning completely. 15 American states ban reproductive cloning, and 3 states prohibit use of public funds for such activities. The Patients First Act 2017 aims to promote stem cell research using cells that are ethically obtained that could contribute to better understanding of diseases and therapies and promote the derivation of pluripotent stem cells lines without the creation of human embryos. Controversies. In bioethics, the ethics of cloning refer to a variety of ethical positions regarding the practice and possibilities of cloning, especially human cloning. While many of these views are religious in origin, some of the questions raised by cloning are faced by secular perspectives as well. Perspectives on human cloning are theoretical, as human therapeutic and reproductive cloning are not commercially used. Animals are currently cloned in laboratories and in livestock production. Advocates support development of therapeutic cloning in order to generate tissues and whole organs to treat patients who otherwise cannot obtain transplants. To avoid the need for immunosuppressive drugs and to stave off the effects of aging, advocates believe that patients who cannot otherwise procreate should have access to the technology. Opponents of cloning have concerns that technology is not yet developed enough to be safe and that it could be prone to abuse, as well as concerns about how cloned individuals could integrate with families and with society at large. Religious groups are divided on cloning, with some opposing the technology as absurd God's place and to the extent embryos are used, destroying a human life. Others support therapeutic cloning's potential life-saving benefits. The cloning of animals is opposed by animal groups due to the number of cloned animals that suffer from malformations before they die, and while food from cloned animals have been approved by the US FDA, its use is opposed by groups concerned about food safety. Where things are going. Lawrence Brody from the National Human Genome Research Institute says the most likely application of cloning in the coming years will remain in animals. The biomedical applications still remain quite difficult, he explained. We just don't experiment willy-nilly when people are involved. Kevin Sinclair, a professor of biology at Nottingham University, believes one of the main applications of cloning in the future will be creating transgenic animals and adding them to the food production chain. Transgenic animals are those that have had a gene inserted into their genome. This process has the potential to eradicate diseases by rewriting genes related to diseases, but this also comes with its own ethical issues, and in Europe, for example, there is a ban on introducing cloned animal products to the food chain. And these are the works cited I used to research for cloning.